Right. So let me start my third topic in this particular unit, which is about antibodies. So antibodies, when it comes to that, we'll have to understand that they are proteins that are developed in our bodies usually, which help in fighting the different kind of the antigens that are present.
Excuse me, ma'am.
Yes, no?
Sorry for the interrupt. Once I'll repeat your number. Can you check it
I have
once,
put
ma'am?
it. I have put it over there. You can just see in the chat box.
Okay.
It is there in the chat box. Yes,
Okay, ma'am.
immunology, first part, antibodies. Let me just, um, yeah. Now let me share my screen with you so that you'll be able to see. Right. So the basic definition of antibiotics, and we'll look into the structure of antibodies and what exactly are the different kinds of antibodies that are present inside our body, what are immunoglobulins, and the different kinds of antibodies that are present, and what are the antigenic determinants that are present in immunoglobulins, and in case, if at all, there are any kind of abnormal immunoglobulins that are produced. So first you can see that it is a glycoprotein, meaning it has a carbohydrate moiety along with a protein moiety as well. So it's a glycoprotein and it is produced by B cells, especially plasma cells. So a B cell origin, like I told you, in the birds, it is Bursa fabriceus, but we do not have that. We have the thymus glands, in fact. So these, the B lymphocytes that are present produce two types of cells, which is memory cells and plasma cells. Memory cells help in recognition of the antigen after the, like what do you call it, uh, exposure to antigens basically. Whereas B cells that is activated once are the plasma cells that produce the antibodies in response to the appropriate antigen that is produced. So when we do serum electrophoresis, that is, we have a process called as electrophoresis in which we can separate different kinds of proteins. So you can see that our serum albumin, when we do the electrophoresis in the blood, we get to see that serum albumin is the protein that is present. And you have different kinds of proteins which are known as alpha, especially in the kind of globulins class, alpha, beta, and gamma proteins. So this is the amount of protein that is present and the mobility based on the molecular weight that is present basically. So all the antibodies are present in the gamma, that is the gamma globulin fraction. That is the reason why they are known as immunoglobulins, as they react immunologically with the antigen, forming an antigen-antibody complex. And you can change the word antibody and immunoglobulin because it is an interchangeable word. And you can see that around 20 to 25% of the total amount of proteins that are present in our serum. Serum is the straw color fluid that is found inside the blood when the blood is taken, and then it is centrifuged, and based on the density separation, when you do that, you'll get a straw colored fluid that is present in the blood. This is the serum. This usually has the different kinds of components such as proteins, glycolipids, and other components. And when you see that in the proteins that are present in the serum, 20 to 25% of these proteins belong to your immunoglobulins. Now we have 5 classes of immunoglobulins when you get to see. So first is IgG, IgE, IgM, IgA, and IgD. So when you get to see, let us just have a small introduction to the different kinds of antibodies that are present. Like I told you, 5 antibodies are present, or immunoglobulins are present types. And the basic structure, when you look into it, of an antibody, you have a Y-shaped. It is a heterodimer. It has 4 polypeptide chains. 2 identical chains, which are light chains that are present over here. These are the light chains. And these are the heavy chains. Based on the molecular weight, these are determined. So each 2 identical heavy chains have a molecular weight of nearly 50,000 daltons. Identical light chains have 25,000 daltons each. So this is known as your antigen binding site or fragment, FAB. This is the variable region. This depends upon the kind of antigen that is being bound to. So this will keep changing. This is the amino end of the antigen. See, it's a protein as such. Like I told you, it is a protein. So when you have a protein, you will talk about an amino end and a carboxy terminal end. So the amino end is over here at the variable region and the carboxy end is over here. And this is known as your fragment of crystallization, and this is a constant region. This yellow colored part is the variable region that is present. And now we have four heavy chains and light chains that are bound together with the help of disulfide bonds. Wherever you see, there are this is a disulfide bond, this is a disulfide bond, this is a disulfide bond, and you have a hinge region which will help in giving a flexibility to the antigen. I mean, the antibody. And now there are five, depending upon the kind of H region and the light chain. You have five classes. So when you see the H chain, like I told you, five classes, and they are distinct, they are not identical, and each is having a particular designation given to it: IgG, E, M, A, D, and also at the same time, these have a Greek terminology: gamma, alpha, mu, and epsilon as such. And then these are the light chains that are present. Light chains are known as kappa and lambda. See, this is the light chain that is present. So it is known as the kappa chain and the lambda chain. And this is named after the scientists Korngold and Lapari. Human beings' light chain is around 60% kappa and 40% lambda. And both of them have either kappa or lambda, but never will you find both kappa and lambda chains together. So like I told you, G is for gamma, A is for alpha, M is for mu, D is for delta, E is for epsilon. These are the types of heavy chains in the, the IG classes. And then again, both heavy and light chains are of two classes. Or they have two regions. One is the variable region. You can see over here, you can see this is the variable region. And this is the constant region. This is the light chains. These are the heavy chains that are present. This whole thing is the light chain. This whole thing is the heavy chain. And this yellow region is the variable region. This will be constant in nature. So this is constant region. This whole thing is constant region. And this is the variable region that is present. And depending upon what kind of amino acid is present, this is again dependent upon the antigen that is going to be coming into place. So in the variable region, first 10, that is the first 110 amino acid residues are the variable heavy and variable light regions respectively. And this is where your antigen binds to the antibody. So some zones, they are also known as the hotspots. So you have a higher amino acid change that is present. And you also have a complementarity determined region. So how much of it? It's known as the CDR region that is present. So that it is going to go and attach itself to the antigen binding site. So you have around 3 hotspots in the light and 4 hotspots in the heavy chain region also. So they are usually present over here. You will find a complementarity determining region over here. We will come to it in some other time. I'll show you that later. But then this is how it is going to be. And then we have a paratope. So this area is known as the epitope. This area is known as the paratope. I'm sorry, that is the site of hypervariable region, which actually makes, so epitopes are present on the antigens. On the antibodies, you have the paratopes present. And in the constant region, what is going to happen is you have around 104 amino acids in length in the light chain and around 330 or 440 amino acids that are present depending upon the immunoglobulin that is present. IgG, IgA, IgD have around 330, whereas IgM and IgE have around 440 amino acids that are present. And they are almost uniform in nature. So now this is how it is going to be. You can just have a look at it. This is the heavy chain variable region. Heavy chain, I mean light chain variable region. So this whole part is going to be variable in nature. And this is where it is antigen binding site. Now this is the heavy chain. This is the heavy chain, light chain, light chain. So this is the hinged region and there are disulfide bonds present everywhere. And this is the constant region. In this you have around what do you call it? Again, you have disulfide bonds. So this is how it is going to be a loop-like structure. 60 amino acids that are present. You have an that is present in an intra-chain disulfide bonding. Light chain has around one variable light chain and one constant domain. You can see a Cl, and then heavy chain has one variable heavy chain region, that is your VH, and one, or depends, again 3 to 4 constant domains that are present. So in the hinge region also, in the heavy chains, what is going to happen is gamma, delta, and alpha. You have a junction that is between your CH1 and CH2 regions. And this is going to be rich in proline and cysteine. So cysteine also has disulfide bridges in them. It is a sulfur containing amino acid. So this makes it flexible in nature, so that it can assume a different position based on the kind of antigen that is present. And so that also what is going to happen is it will help in the antibody go to the antigen and bind to it. So, but the thing is, this disulfide bridges are prone to heat changes, and at the same time, it is going to be, what do you call it? Uh, I'll have to tell that, you can use certain enzymes which will digest these particular hinge region and the disulfide bridges. So, for example, in case you use papain, papain is the enzyme that is obtained from papaya, especially raw papaya. So this will be able to cleave the, meaning cut the disulfide bridges in the hinge region. And this is how you're going to get the fragment FC fragments after papain digestion, as you can see. So you'll get 3 fragments, 2 FABs, that is fragment antibody region, where it is going to be holding the antibodies, I mean the antigen. So you'll get 3 of those, and then you'll get 1 FC. This is the fragment of crystallization. And then if you use beta mercaptoethanol or mercaptoethanol, you're going to get 2 heavy chains and 2 light chains altogether. And in case you use pepsin. When you use pepsin, which is another enzyme that is present usually inside the, it is a digestive enzyme, you're going to see that, you're going to get a completely 2 different kinds of fragments. So now when it comes to this, let us just have a look at the antigen binding site, which is also known as fragment antigen binding region. So its primary function, as usual, we know that it is going to go and bind to the antigen. So it has a variable region, like I already told you, variable heavy, variable light region, so that it will be able to bind specifically to the respective antigen. So valency of the antibody is the number of FAB regions it is having. And we also have a effector function, which is the fragment of crystallization region. So this area is known as the fragment of crystallization. These are the FAB regions that are present. And so they are used for effecting after the antibody binds to the antigen. The region of fragment of crystallization has a function to do either binding to different cells or else perform a function called as complement fixation, which helps in finally digestion or killing of the respective antigens that are present. Mechanics. So let us just have a look at these different classes that are present. All right, so we have 5 types of heavy chains. So because of that, we have 5 classes of immunoglobulins. So it's immunoglobulin A, GEMAD. And you can just have a basic look at it. You can just see that it is a monomer. IgG is a monomer, whereas IgA is it's having 2 forms. One is a monomeric as well as a dimeric form. And IgM is a pentamer, or it can also exist as a monomer. So pentamer means that, two, a dimer of IgA has two IgA particles, or like, two immunoglobulins attached together. Pentamer also has five IgM together, forming a pentameric structure. And then you have IgD and IgE that are monomers. So you have a different valency. A monomer has a valency of 2 because of two fragment antigen binding regions. Dimers have 2 to 4, and monomer or pentameric forms of IgM has either 2 valency or 10 valency, whereas IgD and E have 2 valency each. Other chains are not present, but IgA has a J chain or a secretory component that is present. Even IgM has a J chain. Then you have different kinds of subclasses present. The most important ones in the IgG include your subclasses IgG1, G2, G3, and G4. So you have 4 subclasses. Monomeric for like an IgA has A1 and A2 classes subclasses, whereas M, D, and E do not have any other subclass that is present. And when it comes to the molecular weight, obviously you will see that IgA and IgM have the highest molecular weight depending upon their monomeric or the dimeric or your pentameric forms that are present. How much amount of IgG and or these immunoglobulins respectively are present inside the serum? So you get to see that around 9.5 to 12.5 happens to be IgG. IgA is around 1 to 3 or 2 to what do you call it? A2 is around 0.5 and M is around 1.5 and IgD is around only 0.03. IgE is around only 0.003. So the least amount of IgE that is present is always IgE. And percentage, if you see also, highest is around immunoglobulin A that is present. Immunoglobulin G, and then A, and then later it will be like, IgM, D, and E. Half-life also, whenever there is an immunoglobulin production, you can see that IgG stays inside the serum for nearly 23 days. And at the same time, it is a debatable question, but it is going to stay the most. Whereas it is IgE which is going to have the least amount of half-life that is present. And how much of it is being produced daily? Intravascular discussion, all this have been given over here. So we'll just have a look at it later. When it comes to the we'll just have a look at these. So when it comes to IgG is one protein or immunoglobulin that is going to be the highest having percentage in fat. It can cross the maternal placenta. It will help in complement fixation also activates. There are two pathways in the complement fixation. One is the classical pathway, which is going to be activated once this the Fc region is bound, or the fragment antibody regions are bound to the antigen. And if you see, Ig1 and Ig3 are going to have a phagocytic action. They help in neutralization of toxins. So we'll just have a look at it later. Now let us just have a look at IgM. Has the highest molecular weight. This is the monomeric form. This is the pentameric form. So there are 5 immunoglobulin M which come together forming the pentameric form. So you can see this is the J chain which joins the different monomers together to form the pentameric fragment. So where is it present? It is because it is having a high molecular weight, it is usually not present in the different kinds of secretions. It is present only as an intravascular, so in the vesicles. As an intravascular component, it is present in the monomeric or the pentameric forms. What are the functions of it? You can see that it is present whenever there is any kind of acute infection present inside the body. Also, it's being released as a primary immune response antibody. It will help in activating either the classical pathway or the other pathway that is present inside the body. And then at the same time, it has 5 FC regions that are present. You can see 1, 2, 3, 4, 5. So it has a very important component, and in the sense it has a multiple complement binding sites, because of which you're having a very enhanced immune response that is taking place, and it is usually present on the B cell surfaces for antigen binding. It helps in something known as opsonization, where it acts as an opsonin. So what happens is it is going to bind to the antigen. Then this whole entity, which is because of its highest molecular weight, it is going to be easily recognized, and then you'll have the different anti how the antigen is broken down because of the different kinds of complement fixation pathways that are taking place. And also you can see that it is the first antibody to be synthesized in the fetal life, and in indicator in the any kind of infection that is present in the uterus, intrauterine infection. And it also helps in protection against the different kinds of intravascular organisms, such as those organisms that are present inside the bloodstream. So any kind of organisms that are going into the bloodstream, you have your for example, against your malarial parasite, whatever. So you have some amount of protection that is there. Your IgM is going to be released, and it is 20 times more effective in the bacterial agglutination as compared to your IgG. Now let us see the IgA. It is the second most abundant constituent that is present, and it is around 10 to 15% that is present, and it has two classes, and they are IgA1 and A2. We also have two types of serum your antibodies, which are IgA, which is serum IgA, and secretory IgA. So secretory IgA is actually present in our tears also. Serum IgA is present inside the serum, that is the blood. So this starts expressing on, or it influences the immune effector cells, so that you'll have different kinds of functions. This is antibody dependent cell cytotoxicity and regranulation of the immune cells. So what happens is basically you will have uh, um, the cytotoxic. There is something called as your mast cells and basophils. So they have different kinds of cells that are within, which will be released upon any kind of uh, let us say infection and allergy. So that is when your histamines, bradykinins, and then your prostaglandins and other cells that are released from the mast cells, and these come and act at the site of infection. Now, when it comes to your that is when your IgA is going to trigger those cells. So when it comes to secretory IgA, it is dimeric in nature. Serum IgA can be pentameric also in the form so this is the pentameric IgM. This is how a dimer of IgA is present. So you have a J chain also over here. So you have a secretory component. So where is this IgA present? It is present in milk, saliva, tears, etc. And at the same time, what does it do? It gives us the local immunity that is present. And this is going to be effective against different kinds of bacteria, such as your Vibrio cholera and Neisseria gonorrhea or Salmonella. So they cause different kinds of diseases, such as your gonorrhea or cholera or your typhoid. So it is slightly effective against it. And at the same time, it is present in the breast milk. So secretory IgA is one of the richest sources that is present. This helps in giving the infant gut the protection against the different kinds of pathogens that might be occurring. IgE that is present has the lowest serum concentration. So it is also having the shortest half-life. I have already dealt with it or discussed about it. So where is it present? It is present on the surface of tissue cells. What cells? Mast cells. These mast cells release different kinds of components that are present basically, that are, like I told you already, histamines, bradykinins. So it is a type of, it is the most important type of uh, cells that that are secreted during the type 1 hypersensitivity, that is your allergy. There are a lot of people who are having allergy to certain substances that are present, be it pollen grains, or be it dust, or be it peanuts. So, whenever such kind of exposure is happening, IgE goes, binds to, or mediates the type 1 type of hypersensitivity. Now, whenever there is hypersensitivity, what happens is the mast cells are going to be triggered. So because of which, whenever mast cells are triggered, you have histamines, bradykinins, prostaglandins, then you have different kinds of interleukins, and then tumor necrosis factor, and then proangiogenic factors that are all released by the, especially histamines, so which are released by the mast cells. So this, what happens is this, because of which it will it will cause your allergic reactions that is going to come, or they're trying to counter that particular allergy. In case of any kind of helminthic infection, that is, helminths are your platyhelminthes, nemathelminthes are your invertebrates that are present. So you have liver fluke, lung fluke, and tapeworms. So such kind of helminthic infection, you're going to have allergic reaction symptoms. So it is a marker, you can call it. Now we have one more immunoglobulin, that is IgD. It is present on the membrane on the surface of B cells. So it's a B cell receptor along with the IgM. So we have seen that when it comes to the antigenic determinants of IgG, we all know that antigenic determinant, meaning that it's a what do you call it? It's a glycoprotein. So where is it that, based on the antigenic determinant, you can divide the immunoglobulins, these proteins, into different types: isotype, idiotype, or allotype. Isotype means all the based on the heavy chains. See, this is the heavy chain, right? So in this heavy chain, you're going to see that this all of it have they are going to change only in the amino acid sequences. So all the 5 immunoglobulins, they have different types of heavy chains based on amino acids. They are known as isotypes. So only differences in the amino acids, and the constant region of the amino heavy chain will be changed. Idiotype is wherein, in the variable heavy region and variable light region, whatever is there, it is going to have one uh, area where it can recognize the antigen. So such antigenic determinants are known as idiotypes. And when you see all of it, they are known as idiotopes. Now we have something called as the allotypes. So what is happening? Within the same class, for example, IgG is divided into 4 subclasses. So these subclasses are going to be having, differences in the allotypes. Anti-allotyped antibodies found in the sida also have the rheumatoid arthritis factor that is present. So this is about the the different kinds of antigens and the antigenic determinants that are present. Now we have certain abnormal proteins or abnormal immunoglobulins that are present. When are they released? So you have something called as the Benz-Jones proteins. In the Benz-Jones proteins, you have, it is present especially in the myeloma, basically. So what is going to happen is, in different multiple myeloma, you have more amount of this light chains that are going to be produced, more amount of immunoglobulins that are produced, especially the light chain region. So you can call it as a cancer of the plasma cell, which is going to produce more amount of light chain. So what is going to happen is, you can see that in the serum and in the urine. You also have something called as the Waldenstrom's macroglobinemia, which is also known as the B-cell lymphoma. So what is going to happen is abnormal production of the immunoglobulin M. At the same time, we have a heavy chain disease, which is known as the cryoglobulinemia. So blood has a factor which is known as cryoglobulin, which is going to be moved. And this is associated with, again, multiple myeloma. So myeloid cells, whatever are there, are going to be more. Your T cells, B cells, these are going to be more. So your plasma cells. So when they increase in abnormal amounts, it is known as multiple myeloma. And then it is also seen in hepatitis C infection, wherein your liver is affected. So this is about your abnormal immunoglobulins. Just give me one moment. I want to share some other slide. So just a recollection of it. We'll just have a look at it. Like I told you, the definition of immunoglobulin, different you have the antigen binding and the effector functions. So basic immunoglobulins are heterogeneous in nature and myeloma proteins are homogeneous immunoglobulins. So these are the variable light, constant light, variable heavy. This is the heavy chain. This is the light chain in the blue color. So variable light, this is constant light. Variable light, constant light. Variable heavy, constant heavy. So you have three constant regions, only one variable region that is present. You have intra and inter disulfide bond chains. So like I told you, once this is done, this is the hinge region and you have different types of domains in the oligosaccharides. This is the this is how the IgG looks like. So variable structure of the hypervariable region also depends and based on the complementarity determined region also. Papain digestion, you're going to get three parts. So 1, 2 and 3. And valency is wherein you know how it is going to be binding to. So 1 antigen, 2 antigen. So your valency is going to be 2. And like I told you, this is the second part. Whenever there is a pepsin digestion with the disulfide, like the hinge region also, it is going to be separated. So these are the different human immunoglobulin classes: G1, G2, G3, G4 subclasses. I mean, and then alpha 1, alpha 2. This is the kappa and the lambda chain that is present in the variable light chain types. Again, lambda has 1, 2, 3, 4 types. And like I told you, when you come to the nomenclature, it is IgM has a kappa light chain. IgA1 has a lambda 2 type of variable light chain. So this is the variable light chain. This again is of two types, like I told you, kappa and lambda. So when it comes to IgG, like I told you, complementarity fixation is done by IgG. Placental transfer takes place by Ig2. These are slightly important points which you'll have to remember. Phagocytic, like an opsonization process is there, and the K cells, when they are triggered, you have an antibody side-dependent cell cytotoxicity. So this is the IgM pentameric structure, and C is third highest in the serum, and it is the first made immunoglobulin in the fetus. And like I told you, see here, fixation of C1 by the IgG-MM antibodies. So no activation, but there is activation over here because of the different kinds of CDR regions that are present. So this is the FAB-FC region. So this is how the cell and B cell antigen receptor is going to be as such. And when it comes to IgA, you can just see the secretory is also. So this is one thing. Now we will just have a look at the T cells and the functions of immunoglobulins that are present. So far we have seen the antibody structures and now we will see the functions. So we have stem cells inside the bone marrow. These stem cells develop into B cells, T cells, and all of these. So they are having different kinds of precursors. Basically, you have lymphoid precursor and myeloid precursor, erythropoietic stem cells that too. So these give rise to lymphocytes, granulocytes, and agranulocytes. So when it comes to that, after maturation, we go to the different micro or lymphoid organs, that is the spleen or the lymph nodes that are present in different parts of the body. Then they they get activated. So each B cell that is present inside the body is going to produce antibodies. So when you see the clonal selection, this is the antigen that has a receptor on it, and antigen receptor is going to be over here. These are the antigens that are present. This is the B cell that is there. So whenever there is an antigen that comes, it goes, and these are going to be attached to the antigen receptors that are present on the antibodies, which is already attached from the Fc end to the B cells. Then you have cell proliferation, cloning of the plasma cells or memory cells, and the antibodies are circulated in the bloodstream. So whenever we have a humoral type of immunity, what is going to happen is we have a programmed cell death is taking place. So what is going to happen is the cell is going to die. So you have around 100 million lymphocytes that are being produced every day. So B cells that are not going to encounter any kind of antigen will die because there is no antigen present. But the necessity for production is there. So they said if at all there is any kind of infection present, that is when they undergo activation, and again they undergo the phagocytosis. So many virus infected cells also, whatever are there, will undergo apoptosis, that is programmed cell death. The cell will die from within itself. So what is going to happen is even the virus is going to be killed. So you have clonal selection and detection in that, wherein you have your B cell and the T cells that are going to be detecting it. Now we have something called as the helper T cells. You can see that you have class 2 and class 1 major histocompatibility complex. So what is going to happen is this is the helper T cell. This has a CD4 area, which is actually going to identify this as helper T cell. And this is your macrophage that is present over here. And this is the bacteria. So what is going to happen is your antigen fragment is over here. This is the T cell. So this is going to activate. You have one more enzyme called as the interleukin, which is going to be released. This will activate the T helper cell, because of which when that will happen, antigen fragment is over here. The bacterium is being digested. You have release of interleukin 2. So because of that, what is going to happen is cell-mediated immunity is taking place. At the same time, memory cells are going to be there. And at the same time, you have antibodies also released. So like I told you, we have different types of T cells. We have suppressor T cells, we have cytotoxic T cells, we have helper T cells. So TC cells are the cytotoxic T cells which destroy the target cells, kill the antigens on the surface of all the cells that are present, and then they kill the host cells also. So what enzyme is going to be released? It's a perforin. So there's a pore in the target cell. So what is going to happen is cell lysis, because of which there is apoptosis also taking place. Now, this is how it is going to take place. Now, we have a MHC class 2 molecule that is over here. And this is the T cytotoxic cells. This is the antigen fragment. You have perforins that are present, which are going to be released out. So, this infected cell with the, what do you call it? Different types of whatever infection is there, bacterial or viral, is going to be attached to the TC cells having through the MHC class 1 molecules. Perforins are released, so holes are made, and loss of ion and water, or depends upon the kind of oxygen, the gradient of the ion. So because of which ion and gradient might either enter or come. And then because of all of this, what is happening is infected cell is going to be lysed. So different kinds of immunoglobulins, we have already seen antibody protection also, how we have a humoral response. So what is happening is bacteria, and this is present with T-dependent antigens basically. So this is the class 2 MHC protein, and this is the macrophage that is present. This is going to become an antigen presenting cell. So this is going to come out, and then over here you have a T cell receptor and the helper T cells. Because of which what is happening is B cells are going to be triggered. You have other factors which are like interleukin and other cytokinins that are going to come out. They help in activating the helper T cells. Because of which what is going to happen? Now this will lead to the cloning of memory B cells, wherein you have against this antigen memory B cells being formed. plasma cells and antibodies. So first thing is what is going to happen is remembrance of this antigen so that antibodies can act off it on it upon exposure later. And then second thing is antibodies are released so that this particular pathogen, whatever is there, has to be killed. So when you see the immune responses, we have B cell, which is the humoral, and we have the cell mediated. Both of them have a very critical and important role inside the body because both are depending upon the kind of antigen that is going to be exposed will be triggered. So what is happening? First exposure of antigen. So what is going to happen is this antigen, foreign particle, is going to be eaten away by your macrophage. Then this macrophage with the antigen becomes an APC, which is your antigen presenting cell. So this will go call upon the helper T cell, which will stimulate the B cells as well as the cytotoxic T cells. These B cells give rise to memory cells and plasma cells. These plasma cells secrete antibodies so that this macrophage plus antigen is going to be killed. Over here, if you see this helper T cell stimulates the cytotoxic T cells, which will activate the cytotoxic T cells, and because of which what is going to happen is cell lysis is going to take place. At the second time, what is going to happen is whenever there are memory T cells, they will remember that, okay, this is an antigen which has already been exposed earlier. So we will have to kill it because we do not want that entity inside the body. So again, against the second exposure, memory cells will work, release plasma cells, or they will release the activated cytotoxic T cells completely so that the antigen, whatever is there after the first dose and the second dose, is going to be killed up by the antibodies which are secreted by the plasma cells as well as the activated cytotoxic T cells. So both cell mediated response as well as your humoral, that is your antibody mediated immune response, will take place. This is the whole principle in the case of vaccination. That is why in the case of vaccination, you're going to give a first primal dose. That is when it will cause a little bit of fever and other kind of symptoms, wherein, you have purposefully entered or injected a person with antigens. So there is a humoral response that is going to take place and then a cell mediated response also after a few days. Second time, you're going to give a bigger dose. This bigger dose will trigger the immune response immediately because it has activated the memory cells that are already there circulating inside the bodies. So this is how your immune, humoral and cell-mediated response will be taking place. Now we have applications of monoclonal antibodies that I'll have to deal with. So we'll just have a look at it briefly and then I'll go to the second topic right now. So what are antibodies? We have seen that monoclonal antibodies are because of one single type of cell. They are known as monoclonal because all of them come from one single parent cell. Polyclonal, depending upon the cell lines that are present, they are known as polyclonal antibodies and they only have a different amino acid sequence present. So, purpose of monoclonal and polyclonal, see, first thing is it is produced B cell types. Whereas over here, A single B cell clones. What do? But they bind to different kinds of epitopes of different antigens. Whereas monoclonal, only a single epitope is present and a single antigen is going to be present. These monoclonal antibodies bind to them. Antibody class that is involved is your all kinds of isotypes are present. In fact, a mixture of different kinds of antibodies are present in polyclonal, whereas in monoclonal, only one single antibody class is present. So you can see, so cross-reactivity high is there, whereas low in the case of monoclonal antibodies. Antigen binding sites are also going to be different. When it comes to the history, Littlefield had actually tried to develop this by using something called as the HAT medium, hypoxanthine aminopterin thymidine medium, in the year 1964. Then Kohler Milston provided that by fusing normal and malignant, that is cancer-causing cells. So it was in the year 1990 only that Milston produced the first type of monoclonal antibodies. And after that, you had Ehrlich trying to use these, and he described them as the magic bullets. What are the uses? It's single type, so it is identical. It is directed only against one particular antigen and antigenic determinant. It's also produced by your B cell clones of one single parent. So only a single hybridoma cell line is present. So, like I told you, what is it used for? It is seen in diagnosis, research purposes, therapeutic purposes. So, because of which it can actually help in curing certain types of diseases also. So now what is happening is when you're producing monoclonal antibody, you're doing it by using cell lines that are obtained from different kinds of study animals. So you're taking your B lymphocytes or B cells, then you're going to fuse it with the myeloma cells. Then you're trying to grow the monoclonal antibodies. You're going to take this particular fused cell, inject it into the peritoneal cavity of a mouse, mostly mouse or mouse ascites method, and then in vitro culture. So immunization is taking place, antibody forming cells are taking place, tumor cells are taking place. So first is fusion of this particular, and once this is done, this is screened for if whether this particular total fused product is going to form antibodies. Then if it is present, then it is taken, injected into the peritoneal cavity inside the mouse, wherein you have many kind of antibodies that are being produced, which are taken again for cultivation. So this is being done in a medium called as the HAT medium, hypoxanthine aminopterin and thymidine medium. So immunized myeloma cells without your human growth promoting hormone or the TK region, just which should not be there. Then you using polyethylene glycol, which is PEG, and then inside the HAT medium, you're going to put them in the microtiter plates. Then see if at all there is any kind of antigen specificity and types that are produced. If at all antibodies are produced, then it is going to be taken, added to the mouse so that you have what do you call it? So what is happening is earlier you have done that particular process. Then you're going to take that and then you're going to see cloning of the hybridoma cells, then removal, and then supernatant is taken or mouse ascites is taken basically. So immunized animals, spleen cells, then fusing spleen cells with myeloma cells using polyethylene glycol, which is a fusating agent, and then add aminopterin. So this is going to kill the unfused myeloma cells that are present, and then cloning on the remaining cells in the cell plates. Basically, you have a 96 plate titer. In that you can just do, and then harvest this antibodies from the supernatant. So this is how it is done. What are the types of monoclonal antibodies? So you have different kinds from low to high. If you see murine, chimeric, humanized, primatized means for the primates or humans. So this is the chronic therapy. So this hybridoma technology is how we are going to get your monoclonal antibodies present. So why is this method used? See, you need adequate cell lines. We cannot do that in the in other ways. So it is best to do it in the what do you call it, inside a medium which is the mouse medium. Where is it used? Where are the monoclonal antibodies used? To detect pregnancy or diagnosis of different kinds of diseases. For the presence of a substance if it is present, and in case it is present, to quantify it, and then further you can do a Western blotting, in fact, or an immunofluorescence test. And then this is the sodium esterase page. After that, you're going to take this, add on to the nitrocellulose membrane and perform the Western blotting to see whether the proteins are there. And then you will get the immunoprecipitation and affinity chromatography. But then problems are that it is a standard, to see the mouse antibodies, but they are rejected by the human immune system. So that is why you have conjugated monoclonal antibody therapy that is present, binds to the surface of the tumor. So this kills the tumor cells basically. And so obviously it is used for cancer treatment. Side effects, of course, you'll have different kinds of side effects based on the kind of what do you call it, the type of monoclonal antibodies that are being used because it will help in starting the complementarity fixation also. So that is the reason why whenever there is chemotherapy, in chemotherapy you have different kinds of monoclonal antibodies that are used. See, Avastin, bevacizumab, or anything which ends with your MAB is known as your Zilumab. So you have different kinds. So MAB means monoclonal antibody. So this is your bevacizumab, like targets the tumor blood vessels, growth. So what is going to happen is you're going to have a high BP, bleeding or poor wound healing, more of blood clots, etc. So yes, you have different kinds of monoclonal antibodies and the different kinds of side effects that are taking place. So when it comes to the hybridoma technique, we have the Kohler and Winston actually won a Nobel Prize for it. And B lymphocytes can actually mutate into different kinds of tumor cells, because of which you get myeloma. So whenever there is a myeloma cell, what is going to happen is this cell is going to be immortal, will keep on going and growing in the culture. So this kind of activated B cell and a myeloma cell completely, when it is fused together, will form a hybridoma cell. As you can see over here. So same thing has been what do you call it, explained earlier also. So this is how screening of supernatant for the different types of antibody activities is done. And then you have the hybridomas producing one single type of antibody. So senescence is aging which is not taking place in the plasma cells of the immunization. So whenever you have AGPRT and the deficient immunoglobulin deficient mediums, that is when it is going to take place. So you can see chromosomes also here. Normal chromosomes will die after 7 to 10 days, whereas cancer chromosomes, T cells or B cells, will keep on growing in the culture. Whenever you use PEG, which is your fusant, it is going to have the normal lymphocyte nucleus as well as the heterokaryonic structure. So when this is there, sorry, excess amount of whatever chromosome that is not going to stay anymore is going to come out. Whereas in hybridoma, you have the normal T cell or B cell genes, which will grow up like cancer. So this expression after taking place, you're going to see whether any kind of antibodies are going to work. So your B cell hybridoma will produce a monoclonal antibody, whereas a T cell hybridoma will produce interleukins. So you can just see hybridoma cell line in dilution factor. So you have a 9612, right? And this inverted microscope will be able to detect that. So this is about unit 3, that is antibodies. I'll have to briefly talk about what do you call it, regulation of the immune functions, that is your major histocompatibility complex and complement fixation. We're actually running late. Uh, so I'll just give a brief introduction regarding that also. So when it comes to the functioning of immune system, I have already just given you the differences between humoral and cell-mediated immunity. So we'll just have a look at it like I told you. It's going to be a brief one, like so cell-mediated immunity, you have T cells. Humoral immunity, you have B cells. No antibody formation, but you have antibody formed from the plasma cells. And receptors are going to identify the antigens, but as the antibodies that are there, that are produced, are going to identify the antigens. So basics, general, differences are there. Then humoral, like I told you, just, you have B cells that are present in the humoral immunity, whereas in the cell mediated, you have T helper, cytotoxic, and then you have the you have the T suppressor cells also that are produced. They activate the phagocytic cells and phagocytosis takes place. And I want 5 minutes of your class, so please, bear with me. So this is how the visual review is going to be, how the immune system, like I had already given this particular aspect later all like earlier also, but you can just see different kinds of antigens in a body. Whenever there is a humoral response, you have this B cells and antigens come, and then the antibody secreting plasma cells are there, and then the antigen is going to be eliminated. Whereas when it is the cell mediated response, what is going to happen is the TC cells are there. The T cells are going to form the T helper cells and the T cells, cytotoxic cells. You have the involvement of class 2 major histocompatibility complex molecules that are present. So what is going to happen is releasing of cytokines, and then when cytokines are released, and then you have the killing of the different cells that have been altered. So basic. So when it comes to the cytokines, they are mediators having a low molecular weight, and they are potent, and they are present in the body as hormones mostly. They are autocrine, they act on the self cells, paracrine on the neighboring cells, or endocrine, wherein their site of action is away from the site of production of them. And they are highly interactive in nature. They have different functions. So, they go to different cells. So, they are pleiotropic in nature, they are redundant, they are antagonistic in nature. So, like I told you, autocrine, paracrine, endocrine functions. Highly interactive, pleiotropic, meaning, their target cell can be different. They act on the B cells, thymocytes, and mast cells. So whenever activated T helper cells are there, they release interleukin 4 also. They are redundant, meaning they produce different kinds of cells that help in proliferation of different B cells. Synergistic, what is going to happen? They are going to have both will join together your cytokine family. Cytokines include your interleukin, for example, like I told you, and then they act on together. Or else they can be antagonistic. Two types of interleukins are produced. One acts, one stops its action. So this is how you have different kinds of functions that are present. So when it comes to the different kinds of families that are present in the cytokines, you have a hematopoietic family, interferon family, tumor necrosis factor family, and the chemokine family. All right. So when this is there, we are going to have the first thing is your hematopoietic family, which has interleukins 2 and 4 based on the kind of how it looks and how it is present as such. And when it comes to they are all immunomodulators. They modulate how much amount of immune reaction should take place. So they are ligands in nature. So example is like I told you, class 1 cytokine receptors are hematopoietic, class 2 are interferons, class 3 are tumor necrosis factors, and class 4 are your chemokine receptors, which include IL-8, MIB, and different kinds again. And they're all going to have a signal transduction pathway involvement, wherein they are the receptors for cytokines, they are surface component present. In fact, they are single transmembrane components, and they are multimeric also. So they are all going to be involved in different kinds of signal transduction pathway. Your example is your JAK-STAT that is present. They all have INF alpha as a cytokine receptor. This will trigger your JAK and JAK1 and JAK2. So it's a JAK fast. What do you call it? JAK stands for Janus kinase. So this is a signal transduction pathway that is taking place. So this is how it looks like. Need not worry. Usually it is not there in the bachelor syllabus. You'll have to worry about it in the masters. So macrophage, whatever is there, resting T helper cell, when it is getting activated, releases different kinds of cytokines such as interleukins, TNF alpha, depending upon the kind of action it needs. For example, whenever the activated T helper cell is there, if it wants a stem cell, it will trigger IL-3, IL-6, IL-7, etc. If it wants an antigen to be killed, it will activate or release interleukins 10, 4, and 4 to activate the resting B cell. In case it wants to kill or produce plasma cells, it will release IL-6. For clonal expansion, you have different kinds of interleukins. For allergic reactions, it will go IL-3, 4, 10 act on mast cells, because of which you have allergic reaction or release of the mast cell components taking place. So if it has to trigger or your natural killer cells, IL-2 is involved, or TC cells, then you have IL-12 involved. So different functions and different interleukins, which are all immunomodulators. You will have to remember that particular point, which is very important. So different cytokines, different actions, different set of actions that are present. So once this is done, this is how the interleukins will go. For example, this is the pathway that is given over here. You can just have a look at it. It is going and triggering your stat pathway that is there, and then different kinds of interleukins and interferons are also released. So this is the basic function of the cytokines that are present. I'll have to deal with give me a minute or so. Let us just have a look at the major histocompatibility complex. I request you to just spend another 5 minutes, so that we will be able to finish at least a little part of this particular one. I'll complete major histocompatibility complex and then I will stop. The rest of the class, that is your, I have already finished humoral and cell-mediated immunity. I have only cytokines also I finished. I have interferons and the complement system that has to be taught. It is a bigger topic, so I will have to have some time. So today I want to finish your major histocompatibility complex. We have around 11 slides, so just give me 5 minutes. I will be able to do. So what are major histocompatibility complex? You see that there is a virus infected cell over here and an antigen presenting cell. So different kinds of complexes that are present over it. You have class 1, class 2, T cells receptors, CD4 and CD8 receptors that are present on different cells. So major histocompatibility complex is a particular set that is present on the cell surface of different molecules that are necessary to present an antigen to the T cells. So what does it do? It recognizes the self and non-self. So it also helps in the presentation of processed antigens. Now, where are they present? They are present. For example, your major histocompatibility complex 1 and 2 are the 2 types. So MHC1 is present in all the nucleated cells. MHC2 is present on B cells, plasma cells, macrophages, and dendritic cells. This is how the structure is. So you have 3 components here, alpha 1, alpha 2, 3 domains, alpha 1, 2, 3, and this is a beta 2 microglobulin attached. And this is present as an IgG fold. You have a you have sulfide bridges also present. This is the major histocompatibility type 2 that is present. It has 2 alpha chains and 2 beta chains, beta 1 and beta 2 microglobulin that is present. You have a transmembrane segment that is present, which is going to be inside the cell's transmembrane structure. So this is around 45 kilodaltons that are present, and then you get to see that you have 3 alpha domains and alpha 1, alpha 2 interact. This will help in presenting the processed antigen, and then this is usually a monomer that is present. Now microglobulin, which is a 12 kilodalton, is going to be non-covalently associated with the alpha 3 domain. So microglobulin is the only member in the which is a superfamily which is not having a complete linking. Instead, it is non-covalently linked. MHC2 has 2 alpha domains, 2 beta chains, and they are non-covalently associated again, and each has 2 domains, and alpha 1, beta 1 interact. Alpha 2, beta 2 are present, and they belong to a immunoglobulin superfamily. So these alpha 2, alpha 1 and alpha 2 present the processed antigen, which is around having 13 to 18 amino acids. So you have a C here, alpha 1 domain, alpha helix, beta pleated sheets. So you have a cleft that is present. So then what is going to happen is you have something called as the genetic mechanism, which is going to have a huge diversity or the haplotypes, that is alleles linked together. So synteny is the association of genes in a distinct region that is present. So these MHC1 and 2 have polygenicity, polymorphism, and codominance that is present. These are the genetics of MHC1 that is present. Basically, you can just see that whenever there is a syntenic organization, whenever there is a polygenicity, you have MHC2, 3, and 1 types, wherein you have different kinds of regions. And what is going to happen is, what are the gene products? So what is happening is there is no genetic rearrangement taking place. So humans have something called as the DP, DQ, and DR regions, having which are specifically in the alpha and the beta chains of MHC2. What are they? They are the gene products which are alpha beta in present, that are present. So there is no genetic rearrangement. This is going to be their lifelong, in fact, and they are having around polymorphic structures, and they have around 60. So this is all the genetic level of MHC molecules that we are talking about. So now I will stop here. Uh, like you know, and then what you call it, I'm going to continue in the next class or the next week regarding the complement system and the next topic altogether, which is your immune system in the health, which is the different kinds of hypersensitivity. All right, yeah, thank you so much for your time. Thank you.